Hey everyone, and welcome to today's Explainer. So, today we are breaking down a genuinely groundbreaking new system. We're talking about a platform that promises to completely flip how artificial intelligence actually conducts scientific research. If you've ever wondered how we bridge the gap between a chatty, general-purpose AI and rigorous, Nobel-level science, well, you are in the exact right place. We've got a lot to cover, so let's jump right in.
Okay, let's dive into this. Have you ever noticed that while general-purpose AI models are incredibly articulate, they just kind of fall apart when faced with the chaotic reality of modern science? I mean, real scientific workflows are a completely fragmented mess. You've got papers, sprawling code bases, raw data matrices, unformatted protein sequences, and really complex team decisions all happening at once. When standard, chat-based AIs are thrown into that exact environment, they frequently hallucinate evidence. They forget context. It's a bit like inviting a world-class philosopher into a high-stakes wet lab. You know, they're fantastic conversationalists, but they're absolutely terrible at managing the pipettes and spreadsheets.
The core of the problem here is a fundamental mismatch in design. Your standard general-purpose assistants operate on session-based, text-only interactions. But real science, it demands persistent goals that span across days or even weeks. It requires multi-role collaboration among human experts, and most crucially, strict auditable evidence traces. If you make a scientific claim, it absolutely must be backed by a specific dataset, parsed by a specific script, under specific parameters. Standard models simply cannot provide this level of rigorous accountability out of the box. They just weren't built for it.
So enter the solution. Developed by the team at the Shanghai Artificial Intelligence Laboratory, this is SciForge. And I want you to think of SciForge not as just another chatbot wrapper, but as an AI-native, local-first workbench. This is huge because it means your unreleased, highly sensitive data stays totally secure right on your machine. It acts precisely like a meticulous lab manager. It preserves your familiar graphical interface for human judgment, while seamlessly orchestrating things like search, parsing, model routing, and workflow execution as modular services running securely in the background.
What's really fascinating about SciForge's architecture is its translate-then-reason model. Unlike standard AIs that try to ingest absolutely everything as raw text, which we know leads to mistakes, SciForge knows its limits. So step 1, it takes in your native scientific files. But step 2 is the real game changer. Instead of feeding unformatted data directly to a general AI, the system dispatches them to domain expert translators. For instance, say you have a raw FASTA file, which is essentially just text representing a protein sequence. SciForge routes that to a dedicated expert translator called ESM2Text. Then, in step 3, these translators return structured expert observations. Finally, in step 4, the main AI agent reasons over this translated, highly trustworthy data. And if a file format is unsupported, the system is designed to fail safely closed, rather than just guessing and hallucinating an answer.
Now, what's really interesting about this slide is how the interface perfectly captures that lab manager vibe we just talked about. On the left, you're seeing your ongoing live scientific research thread. This is your dialogue and execution history. But on the right, you have this inspectable evidence-directed acyclic graph, or DAG for short. Basically, think of it as an unbreakable one-way flowchart of evidence. This visual sidecar literally forces the AI to show its receipts. Every single claim is permanently linked back to its source assertions and reasoning steps. So as a human researcher, you are never left guessing where on earth an idea came from.
Let's look a little closer at this thread-level evidence DAG, specifically during a target discovery session. This intricate graph maps out 32 distinct nodes, connected by 21 edges. Notice how the visual structure deliberately flows from left to right. It explicitly separates the foundational source assertions over in the left columns from the reasoning nodes in the middle, which then flow into the final claims on the far right. And here is why this is so critical. During this specific audit run, this visual sidecar instantly exposed a fabricated protein structure reference that an agent actually tried to sneak in. Because these relationships are visually mapped out like this, the human researcher could immediately spot the unsupported claim and reject it. The AI does the heavy lifting for sure, but the ultimate scientific judgment stays firmly in human hands.
But science isn't done in a single sitting, is it? Well, this system scales up beautifully from a single thread to a shared team workspace via the project-level DAG. What you're seeing right here is an aggregated snapshot summarizing 4 distinct research sessions. It pulls together 25 evidence records and 16 claims into a massive 45-node macroscopic view. Think about what that means. You have true cross-session memory and multi-role decision governance. A primary investigator can log in and instantly trace the entire lineage of a project's findings across different days, different experiments, and different team members. And this brilliantly illustrates what happens when you let this system run on its own. This flowchart maps out an automated agentic research sprint, and honestly, the scale of it is staggering. Given a simple biological prompt about meiosis, the AI navigated an incredible 132 distinct stages. It moved autonomously from literature triage, analyzing 164 papers, straight through to synthesizing multiomics evidence, where it pulled together massive biological datasets. It utilized AlphaFold 3 for structural validation, and ultimately, it synthesized a living version-controlled manuscript. Across the single run, it executed 199 commits to the codebase, spawned 6 parallel child agents, and generated 9 figures, all while keeping that continuous auditable thread of evidence perfectly intact.
To truly appreciate this level of autonomy, just take a look at this auto-research progress scatterplot. Here, the AI was given a strict 7-minute budget to improve a protein contact prediction model. The horizontal axis tracks the 24 independent experiments it ran, while the vertical axis tracks the primary accuracy metric. Those gray dots? Those are the discarded experiments, but the green dots, along with that rising step line, show the 9 distinct improvements it actually decided to keep. The AI independently explored its own adjustments. It tested different attention layers to see what stuck. It tweaked epoch counts, trying out 800, then 1200, and it optimized dropout rates. It essentially hypothesized, tested, and iterated entirely on its own. It's like having a tireless lab tech running human trial-and-error experiments, but at digital speed. And crucially, it didn't just output a bunch of numbers. It generated hard visual evidence for its progress. These are the contact map visualizations for 3 specific evaluation proteins, labeled 1A3A, 5AHW, and 1XCR, taken right from the 24th iteration of that exact experiment. If you notice the diagonal lines slicing through each square, the upper panels display the AI's predicted contact probabilities, while the lower panels mirror the native true contacts derived directly from the physical structures in the PDB. This direct visual comparison, paired with the red and green keep or discard timeline at the bottom, lets the human supervisor visually verify that the AI's numerical improvements actually match physical reality.
Now, beyond just generating research, SciForge can rigorously critique it. We all know the messy, sometimes painful reality of the peer review process, right? Well, SciForge structures this into a meticulous 6-stage workflow. Starting on the intake side, it digests your manuscript PDF along with the reviewer concerns. It then extracts specific claims, explicitly links them to your tables and figures, and conducts what they call a fragility audit, literally categorizing your claims as supported, fragile, or unsupported. Finally, it decomposes the reviewer comments and generates a prioritized rebuttal and revision plan. It takes a stressful, highly unstructured process and turns it into an actionable, fully traceable pipeline.
Watching this review process actually come to life in the interface is just brilliant. It uses a 3-panel view. In the center, you have your recompiled manuscript PDF. On the right side, you have page-anchored reviewer comments that are tied directly to specific paragraphs. And on the left, you can actually see the agent's live thread, where it executes the code edits and runs compilation checks. It makes the entire path from a specific reviewer concern over to the agent's code edit, directly to the final manuscript change, perfectly auditable and transparent.
The system is also an absolute powerhouse at data integration, which is, let's face it, a notoriously tedious and difficult task for human researchers, who usually have to manually cross-reference dozens of spreadsheets. This chart shows SciForge unifying information across 6 biological layers for a 35-guide dataset. The blue bars show the experimental data measured directly in the lab, like foundational genetic perturbations at layer 0 all the way to final protein abundance, achieving nearly complete coverage. The orange bars, however, represent how the AI autonomously enriched that data set. It actively pulled in annotated metrics from external databases, like UniProt and Reactome, to fill in the missing layers in between. The result is this comprehensive, multi-scale biological dataset prepared seamlessly, saving literally weeks of human effort.
Let's move to and see how this builds into actionable insights. Here we have a 4-panel dashboard visualizing the multimodal evidence landscape, linking those biological layers together. The first panel tracks the cells per guide. The second panel ranks the transcriptome-wide effects, highlighting recurrent response genes, and you can see CCL4 right up there at the top. But the real magic happens when we link this to the third and fourth panels, which show the surface abundance changes for critical immune checkpoints. In that last panel, you can clearly see the dramatic mean negative delta for PDL1, which is a critical protein in immune responses in cancer research, compared to just minor shifts in CD86. SciForge isn't just plotting raw data, it's linking gene expression directly to protein-level immune phenotypes, showing us exactly why this matters for real-world drug discovery.
We see this exact same exempling rigorous evaluation in AI-guided protein design.
Look at these 4 comparative charts for candidate 001 in blue and candidate 002 in orange. The first panel highlights the Boltz-2 verification scores, with candidate 1 actually edging out candidate 2 with a 0.9396 confidence score. The next panel gives us a really beautiful radar profile of the amino acid composition. Then we get to see the crucial trade-off between sequence entropy and structure confidence, which is essentially balancing how unique the design is versus how stable it will be in physical reality. And the final panel breaks down the distinct physical-chemical properties, noting precise charged and hydrophobic fractions. Every single metric is exposed, so the human researcher has everything they need to make the final informed call.
Ultimately, all of this incredibly complex architecture funnels down to a surprisingly seamless user experience in the biology room interface. Notice the 3D ribbon structure of the protein here. If a researcher clicks on the specific ALA86 residue within that 9-VMR structure, the system doesn't just copy plain text into the prompt. It sends a highly structured cryptographic hash, which is basically an unforgeable digital fingerprint, along with the exact file path and a precise residue locator straight into the AI chat. This selection-grounded dialogue forces the AI to check the real, physical file before responding, which virtually eliminates hallucination. You are having a fluid conversation, but one that is firmly grounded in cryptographic reality.
So, to wrap this all up, SciForge fundamentally transforms artificial intelligence. It takes it from being a clever but often unreliable black box conversationalist and turns it into a multimodal, highly auditable laboratory partner. By forcing the AI to translate before reasoning and by structurally tracking every single claim in a verifiable graph, it hands the reins of scientific rigor right back to the researcher. It's a massive leap forward, and it begs a really exciting final question for you to ponder today. If your AI could finally show its work with perfect clarity, what scientific discovery would you chase next? Thanks so much for joining me on this explainer, and I'll catch you on the next deep dive.